integrating PAT technologies in high throughput bioprocess development, or what we call high throughput bioprocess development. So the overarching goal, in our view, of a laboratory or of bioprocess development should be the systematic reduction of uncertainty during the process of design and scale up, specifically on the expected optimal production strategy, and again at minimal costs, resources, and time. So this is our aim, and the work we have been doing in the KIWI-biolab goes always with the objective of building the most efficient possible, so to say, knowledge generation machinery for the development of new bioprocesses, and for this to use all the existing and emerging technologies. So in our view, analytical technologies play an essential role in this high throughput bioprocess development. First of all, because it is evident that the analytical technologies that are used, or that can be used, or that could be implemented in the industrial process will define the properties and the characteristics of this process, meaning what can be done, what can be understood, how tight it be controlled, the quality, and so back and so forth. Secondly, because the information and the insights gained through these technologies in large scale and throughout the experiments, of course, are the main, or the raw source of the information that one can be attained. And if we also consider that the new technologies in data science, machine learning, or what now is roughly called AI, allow us to treat ever increasing sizes of data sets, of course, we can exploit now the fact that we have analytical technologies that generate very large data with not so much information, but these tools can extract it and can make the best use out of it. Last but not least, many technologies are also specifically developed for high throughput and for miniaturized systems, and these have really increased the capabilities of these machines, and I have some examples of this following. So first, we see that the true potential is in the integration of the analytical technologies with all the other experimental devices and also the computational ones. And here, the devices that we use in our lab are mainly liquid handling stations, where we integrate inside parallel bioreactor cultivations. The two that we mainly use is the 2MAC bioreactor with up to 48 10 milliliter bioreactors with steering aeration that can be very efficiently handled with a pipetting robot, where we have the possibility to have bolus additions for bolus-based feeding, or with N-based technology, for example, from Empresso, glucose release, enzymatic glucose release systems for fed batch. We also have, in a similar embedding, the HEL BioExplorer 150 with 100 milliliters working volume, and now also pumps, outgas, probes, steerings, everything that a benchtop bioreactor has. And due to the embedding in robots, but also due to a mobile lab assistant that we have that can go to different devices and move plates from here to there, we can also easily integrate analytical tools. For example, the flow cytometer is integrated and can be directly accessed, but we have also a SEDEX bio-HT analyzer, which is physically separated, and the robot can overcome these problems. We should never forget that in order for all this to work, not only the physical connection is important, but the devices need to communicate with each other. There must be a central place where everything is stored, such that the computer tools that we use, the computational tools, can access the information but they also have to be in such a form that they can understand it, use it, and make the most out of it. And we have been working lately very intensively together with a group of Argentina to develop a very nice workflow management system by which also every step and every decision made by the computers is tracked, and by which we have the full record of what happened throughout a complete experiment, making it first of all traceable, such that if something went wrong, we can go back and see and find the source of the error, and also increasing its reproducibility significantly. But now back to our concern. In these systems like the TUMAC bioreactor system, where we have bolus feedings, we can do exponential, we can induce, we can run for long times. Sorry that the scales are so small here, the axis, but this is 54 hours cultivation, more than 2 days. And as you can see, everything works very well. But here we already encounter some challenges. First of all, the dynamics of the systems, which are also constantly in excitation due to the bolus feeding, and where we have very different time scales with very fast dynamics, like the oxygen that goes up and down, specifically in organisms with high glucose affinity, like E. coli, and on the other side, biomass, which grows slowly compared to the oxygen. We also have in the frequencies very different observations, very different frequencies, because we have online sensors that can track and record every 5 seconds or even lower. And then on the other hand, we have all the upline analytics where the frequency is significantly slower. We typically have, from the most important metabolites, relatively scarce information, because for this we have to take a sample, has to be treated and run through different methods, typically every hour, maybe a bit shorter, but not much, and very often even once a day. And if we go to the most important topic typically in these experiments, which is the product, these are usually much more complicated analytic methods, and often we even need some weeks delay before we can get the results. But analytical technologies can really vary, so to say, even by now I would call them traditional ones or very well established, can really completely and drastically change the way we can run this type of experiments. And here, as an example, just having, not just, but having the presence sensors, spot sensors in the bottom of the mini bioreactors, which can measure oxygen and pH, allows us to run the cultivations completely differently. We have by this high frequency, almost online track of the oxygen. There is a short sensor delay, but we deal with this, and also from the pH. And by this, we can not only, roughly speaking, control, for example, the pH close and monitor what is happening with the oxygen, but if we embed it with tools for like soft sensors and this type of machineries, we can go really far. And here we have an example where, using the pH controller and the base that is added to maintain the set point of pH, and a simple mechanistic model, we can, despite the very limited online measurements and changes in the metabolic activity, we can, using a Kalman filter, a state estimator, for example, or filtering, to be precise, really control and see what is happening in the process. And we can go even further, using the amount of base that had to be added. We can estimate really the biomass concentration throughout the cultivation. Here we see the red line, which is the estimation based on the base added using this Kalman filter, the sigma point, also an extended version. And the samplings that were taken at line show that the method is really very accurate. So by this, we can now, even throughout the cultivation, have a very good estimation of the biomass, for example. If we combine this with the mechanistic models that we develop and we use for more than 10 years now in the chair, we can, first of all, estimate the parameters of these models much better, but secondly, even use them for the optimal operation of the cultivations. And here, very shortly, what we can do, some work from Niels, where using estimation methods like the Kalman filter or moving horizon estimator he used in this case, we can first use all the data of the current that already exists from the cultivation, estimate our current states, define what is the situation, the status of the reactor, and by this, with an optimizer, compute the optimal strategy for the continuation of the cultivation. And here what we can see in this animation is a set of 8 bioreactors with the objective to maximize the biomass, but making sure that the dissolved oxygen tension never drops beyond 20%, such that we can be sure that we had full aerobic conditions throughout the complete experiment. And as you see, the MPC is operating each one of these reactors very efficiently, and the constraints are never violated. So in terms of, going further and trying to put different technologies in these miniaturized devices, we also work a lot on how to combine all the methods and technologies possible to get the best insight possible on the cultivation that is running. And an example of this is the integration we have done of the HL bioreactor, mainly done followed by Lucas, where this BioExplorer 150 has been integrated in a liquid handling station such that it can be sampled and added whatever we want fully automatically. The reactors can run in parallel, in this case 6, because of some spatial limitations. We have pumps, we have probes, we have added also the BlueSense outgas analyzers such that we really have the online measurement of the outgas. We can also have, for example, the MuCheck system to have online OD measurement and added the cell size distribution. And by this, we have really a very accurate or a very good overview of what is happening during cultivation. And if we combine this also added to systems like this 3-reporter plasmid developed in Trondheim, where we can, by this, throughout the cultivation, this is again, I hope these are a bit larger. This is again 25 hours cultivations in parallel, running fully automatically. And we can, for example, compare the effects of continuous feeding strategies versus bolus-based on the cells, not only on production with, in this case, an RFP, red fluorescent protein, to control the specific, to measure the specific production, but also in this case, through a YFP reporter, the ribosome synthesis, and to compare the differences and also the response or the stress response of the cells using another reporter. Furthermore, we also work in now a different setting in integrating the Raman technology with a very nice setup that has been built to be able to get the 8 samples of the pipette and through this multiplex order them such that one after the other can go through the Raman system. And by this, we can also online through the process have the measurements. Everything is very nicely and modular implemented such that each device works with a microservice architecture. And now the steps missing is exploit the data that is being generated with machine learning tools to extract the most out of this spectrum. So with this, I hope I could convince you that despite the fact that robotics, machine learning, automation, miniaturization are significantly speeding the bioprocess development, the integration of analytical technologies is not always given the deserved attention or has not been recognized as a bottleneck for some time, and that the real potential lies within the synergy between all these tools, analytical, computational, experimental, such that we can really speed up and significantly improve the development of novel bioprocesses. With this, I want to thank you. I want to thank the KIWI-biolab, and of course, the chair of bioprocess engineering and the head of the chair, Peter Neubauer. And this video should stop now, and now it should be live and ready to try to answer any questions you might have. Thank you very much.